Hi, my name is Chandler Nielson, and here is my name, clearly shown, Chandler Nielson, April 30th, 2020. And today I'm just going to answer my questions that I was able to work on the Unit 2 case connections. Let me switch over to my PowerPoint here, and we'll begin. So I chose case 1, and it talks about pyruvate kinases and how these things are regulated. There's two parts to this, and later on I'll read kind of the other part to help us understand my second part of my answer. But the first part just says, how does pyruvate kinase, how is it regulated? And we'll kind of go down the list here, and we'll make our way down, and we'll go from there. But pyruvate kinase is regulated through allosteric regulation by fructose 1,6 biphosphate. When fructose 6-phosphate phosphorylates, it produces FBP. This molecule is an intermediate in glycolysis and binds to pyruvate kinases. So after these things bond, these enzymes change conformation. And that change of conformation activates the pyruvate kinases. So there's the pyruvate kinase, it's bonded to this FBP, it is then activated, and so pyruvate kinases are regulated by the concentration of FBP. With higher concentration of FBP comes obviously more activity of kinases and vice versa. If we move our way down the list, glycolysis, pyruvate kinase is the most critical enzyme in that last step of glycolysis. In the step, the glucose is formed into pyruvate. Pyruvate kinase is the regulatory enzyme that interacts with PEP molecules to transfer a phosphate group to ADP. We all know, obviously, that this turns it to ATP and then sent back. This step in glycolysis is a rate-limiting step, and obviously this would show that pyruvate kinase is regulated through this process, through glycolysis. We make our way down, we talk about these metabolites. Another way it's regulated are through these. These metabolites regulate pyruvate kinases by intercellular signaling. These signals are sent to regulate glucose metabolism based off what the cell needs. The cell is then able to know how much glucose to metabolize and control the growth of the cell. In a simpler way, if these metabolites increase signaling, pyruvate's activity is obviously increased, and on the other hand, it's decreased with little signaling or no signaling at all. The next is hormones, and obviously we never want glycolysis and glucogenesis to occur in the cell at the same time. So how this is regulated and how this doesn't happen is hormones can help us not have that problem. For example, epinephrine will increase the activity of gluconeogenesis while turning off glycolysis. As stated previously, with no activity of glycolysis in the cell, this will inhibit pyruvate kinase's activity. Lastly, these covalent modifiers, they also help the cell regulate the activity of these pyruvate kinases. Covalent modifiers help control factors that dictate activation or inhibition of enzymatic activity. When insulin is released in the blood, a chain reaction causes this dephosphorylation. This results in an activation of pyruvate kinases to amp up glycolysis to break down more sugar. The opposite happens when glucogen and epinephrine, and that's why these things, well, obviously with an increase of phosphorylation, there's a decrease of this activity. Now the next part of the question kind of is more in depth, and it's more of a compare and contrast. It talks about this protein kinase M2 and then M1. And it says the isoform M2 is characteristically found in low activity state and is ineffective at prompting glycolysis. The logic seems counterintuitive for a glycolytic tissue. What is the rationale for this mechanism and other pathways that the cell can divert to? So pyruvate kinase M2 and PEP, when in discussion with cancer, usually shows, I would say, a direct relationship. When high levels of pyruvate kinase M2 are found in cancers, we see that there's also a high level of PEP. That being said, cancer's metabolism's main enzyme would have to be pyruvate kinase. This enzyme is able to regulate the expression of the gene that is needed for the cancer's growth. PEP, though, on the other hand, can be synthesized into energy as stated in the first portion of the case study With more energy, the cancer is able to replicate faster, obviously. Stem cells use a different path, though. In these cells, we see an increased expression of the PKM2 by a receptor called MYC and the glucose transporter. RNA splicing is promoted by the MYC receptor. This takes place to let PKM2 be expressed. This is the link to the multiplying of cells. Stimulation of the receptor increases glycolysis and glucose uptake. So wherever there's going to be a high affinity for PEP, PKM2 is formed. This then leads to the formation of pyruvate, and then two PEP cells go through phosphorylation to create pyruvate, and this is what is found in tumor cells. With the levels of PEP going down, the intermediates in glycolysis increase. Then I would say, they're channeled to a pentose phosphate pathway, which just leads to more and more and more cancer cells, tumor cells. And that's found right here, and that's the part 2 of that discussion. So case 2 is a little different, and it just talks about how different tissues regulate the uptake of glucose, and it asks us to show how each one does that. So the liver uses GLUT2 as a transporter to uptake or honestly release glucose rapidly into the body. The liver is insulin independent, meaning the glucose is brought in no matter what the insulin levels are. When the liver secretes glucagon in response to low blood sugar, this activates glucogenesis and glycolysis. These release sugar back into the bloodstream to keep homeostasis. When insulin is excreted and the blood sugar levels are high, this triggers the liver to store glucose as glycogen. With a higher uptake of glucose, the blood sugar level can then return to normal. This is how the glucose is regulated. The uptake is regulated in the liver. Now, skeletal muscle, as we move down, in adipose tissue, they use a different transporter. They use GLUT4. Both of these tissues are insulin dependent. This dependence means that these tissues are stimulated by insulin. The cells in adipose tissue and skeletal muscle will transport glucose into the cell when insulin levels are elevated. Then the last on our list is the brain, and our brain doesn't use those well. It uses GLUT1 and GLUT3 as transporters. Now, during the fed and fasted states, glucose metabolism is unchanged, making the brain tissue more or less insulin independent. But during a fasted state, the liver does produce glucose. This glucose is taken up by the red blood cells and by the brain by glucogenesis. This is crucial so that the levels can remain constant during a fed or fasted state. And that's how these tissues regulate this uptake of glucose. Now, the second part of the question was actually really interesting and fascinating to me. It showed this MRI and the PET scans. And it asks you to kind of compare and contrast and look at it. And it says, how does the glucose uptake in a normal cell compare to those glucose uptake in a tumor cell in the above images? And why would this be advantageous in the invasive tumor? What modifications may have occurred in cancer cells to alter glucose uptake as predicted above? Now, there's these two different scans. We have the MRI and we have the PET scan, and they're fluorescently labeled with D-glucose. The PET scan shows the brain with a large and obvious mass, or does the MRI. And you can kind of see this mass much better, though, with the PET. The highlighted mass is showing the radioactive glucose and the region this glucose is being used in. Normal cells do not take up near the amount of glucose, though, as cancer. This creates great contrast on these pictures and enables us to see where these masses of tissue are. Glucose is consumed at a much higher rate in cancer cells because these cells are reproducing at a high rate and have high energy requirements. Cancer cells do not use the oxidative phosphorylation pathway as their primary source of glucose. Normal cells would use the oxidative phosphorylation as their primary source for glucose. The difference is the Warburg effect is what cancer cells use, and this effect is the fermentation of glucose to lactose. and and cells mostly using glucose for glycolysis. That's what the Warburg effect is. Now, this causes increased uptake and fermentation of glucose to lactate. And looking at the PET scan, this is why we can see that luminescent portion of the mass where the cancer is located. Thank you so much for watching, and I'm grateful for that opportunity. I'm a little nervous, and we're kind of all out of whack here being quarantined, but it was nice to understand how to do these presentations. This is my first time, and kind of get the jitters out. But thank you for your time. I appreciate it.